Hi, everybody, and welcome to chapter 17. We will talk about blood gases, the pH and buffer systems in chapter 17. And this lecture was originally developed by the faculty at Valley, and it was revised by myself, Carolina Vilches. So let's start talking about blood gases and why is it important to measure a patient's acid-base balance. It's important because you want to assess the patients if when the patients are in life-threatening situations. So in this particular lecture, we will discuss the exchange of gases, carbon dioxide, and oxygen together with the body's mechanism to maintain acid base balance. So it's very important for the person or the body to maintain acid base. So you want to do blood gases to determine if the patient has an imbalance in oxygen gas or carbon dioxide gas in their blood. You also want to be able to determine the acid base imbalance, which can indicate a respiratory, metabolic, or kidney disorder. When are these tested? So it would be tested if a patient has difficulty breathing, shortness of breath, rapid breathing, or hyperventilation. It's important to monitor oxygen therapy, and during surgery to monitor the blood's carbon dioxide and oxygen levels. The sample that we use for blood gas testing is collected from an artery, and we get capillary blood from a heel stick, usually when we have baby cases. So let's start with some definitions. It's going to be very important for us to understand these in order to understand these systems in the body that control the acid base balances. So the first one is acid. Acid is a substance that can donate hydrogen ions when it is dissolved in water. This is your basic chemistry lecture. A base is a substance that can yield a hydroxyl ion, which is OH minus. A dissociation constant is an ionization constant K, which is a K value, and it describes the relative strengths of acids and bases. pK is the negative log of ionization constant and the pH in which protonated and unprotonated forms are present in equal concentrations. So the pK um, of a solution is defined as the negative log of hydrogen ion concentration. A decrease in 1 pH unit represents a tenfold increase in hydrogen concentration. So that's usually how it works. A decrease in 1 pH unit represents a tenfold increase in hydrogen concentration. And a buffer system or a buffer is a buffer system or a buffer is a combination of weak acid or weak base with its salt, with their salts. A system is basically a system that resists changes in pH. Okay, so why do we measure in the clinical lab? We measure the pH, which is a measure of hydrogen ions. We measure PO2, which is the partial pressure of oxygen, CO2, which is the partial pressure of carbon dioxide. We also measure O2 saturation, which is the oxygen saturation, and that is done by doing a calculation of how much oxygen is bound to hemoglobin and available to be carried through the arteries. So O2 saturation is how much oxygen is available to bind to hemoglobin. We can also measure bicarbonate, HCO3, bicarbonate. So these levels, pH, PO2, CO2, O2 saturation, and HCO3 or bicarbonate are going to be measurements that are going to be put into calculations to define certain acid-base imbalances that we'll talk about a little bit later. So if there are any abnormal results in these measurements above, you can find that there may be issues with the body not getting enough oxygen or the body not getting rid of enough carbon dioxide. So remember, we take in oxygen and we get rid of carbon dioxide. So that has to be in balance in our body, in hemostasis. We could also see improper measurements of pH, PO2, POCO2, oxygen saturation, and bicarbonate when there is a problem with the kidneys, when there's a problem with kidney function. So these are reference ranges from your book. You have your normal ranges for everything that we measure in terms of blood gases, and that's pH 7.35 to 7.45. That is a reference range for the pH. Partial CO2 is measured in millimeters of mercury, which is usually a unit that we use to define gases, blood gases. HCO3 bicarbonate 22 to 29. Total carbon dioxide content, it's measured in millimoles per liter, 23 to 27. Partial oxygen millimole per liter. Saturated oxygen is a percentage which should be greater than 95, and oxygenated hemoglobin should be, it's also a percentage and should be greater than 95. All right, so let's talk about the maintenance of hydrogen in the body to maintain acid-base balance. So it's very important that the body maintains hydrogen ions in the body. The normal concentration of hydrogen in the extracellular fluid can range from 36 to 44 millimoles per liter, which would be equal to having a normal pH, which is 7.35 and 7.45. But the body produces much greater quantities of hydrogen. So this maintenance of pH happens through the action of the lungs and the kidneys. So the lungs and the kidneys are going to regulate hydrogen function or hydrogen concentrations in the body. So the body controls and excretes hydrogen to maintain regular pH levels, okay? So when there is an imbalance between the rate of acid formation, which is hydrogen ions, and excretion, which is when you excrete the hydrogen ions, these imbalances can lead to certain alterations in the body. So a patient can experience consciousness, alterations in consciousness, neuromuscular irritability, tetany, which is sort of like muscle spasms, coma, or even death. So it's very, very important to maintain the balance levels of these. So there are certain things to remember. Again, as I mentioned before with the definitions, the dissociation constant is the ability of acids and bases to dissociate in water. pKa or pK is the negative log of the dissociation constant. So it is the ionization constant. If the pH equals the pKa, or the negative log of the dissociation constant, then the solution is in equilibrium with protonated and unprotonated species, which are all present in equal concentrations. So if the PA equals the pKa in the calculation, that means that the solution has an equilibrium, an equal amount of protonated and unprotonated species. So when we talk about acid-base imbalance or balances, we refer to these issues as in acidosis or alkalosis. So acidosis is when the pH level is below the reference range, which would be less than 7.35 or 7.34. And alkalosis is when there's alkaline and the pH level is above the reference range, which would be greater than 7.44 or 7.45. It is also important to note that buffer systems exist, and a buffer, as I said to you in the definition slide, is a weak acid and its salt of its conjugate base, and it resists the change of pH upon adding an acid or base. So it'll always resist the change in pH when you add an acid or a base. The effectiveness of the buffer will depend on the pKa, on the pKa, which is the negative log of the dissociation constant or the ionization constant of the buffering system, and it will depend on the pH of the environment in which it is placed, the buffer systems. The buffer systems regulate hydrogen ions. The lungs and the buffer systems are the body's first line of defense against extreme changes in hydrogen concentration. So basically, all buffers consist of a weak acid and its corresponding salt or conjugate base, right? There is one important buffer in the body, which is called the bicarbonate carbonic acid system. This has a low buffering capacity, but it is still important, and it's still a very important buffer in the body for three particular reasons. With this buffer, the carbonic acid dissociates into carbon dioxide and water, which then allows carbon dioxide to be eliminated by the lungs and hydrogen as water. Also, this bicarbonate carbonic acid system is very important because the changes in carbon dioxide modify ventilation or the respiratory rate. And lastly, HCO3 minus or bicarbonate concentration gets altered by the kidneys. There are other buffer systems that are important as well, such as the phosphate system and a plasma protein buffer system. And also, it is important to keep in mind that hemoglobin plays a very, very important role in buffering carbon dioxide as it is transported to the lungs for excretion. So hemoglobin here plays a huge role. Okay, so the measurements of components in this bicarbonate carbonic buffering system will provide us information that regulate production, retention, and excretion of acid and bases. There is a particular reaction that goes on in the body. You have the CO2, the carbon dioxide, plus the water, which will lead to carbonic acid and hydrogen ions and bicarbonate. So basically, during cell metabolism, the cell metabolism produces carbon dioxide. Carbon dioxide then diffuses into the blood. It then reacts with H2O or water to form carbonic acid, which is H2CO3. This then dissociates to form hydrogen and bicarbonate ions. Bicarbonate is HCO3 minus. So what happens is in the lungs, the reaction is reversed to form CO2 because you want to excrete CO2 in the lungs. And so that's basically the reaction. So CO2 diffuses into blood, it reacts with water to form carbonic acid, and then dissociates, and then this dissociates to form hydrogen and bicarbonate ions, which is HCO3. Okay, so again, as I said before, the lungs and the kidneys play a huge role in the regulation of extracellular pH or the regulation of hydrogen ions. The interrelationship of the lungs and the kidneys in maintaining the pH is shown